Let's talk about the procedure to prepare the template for the DNA sequencing. इस सारे प्रोसीजर का जो एंड पे हमारे पास रिजल्ट आएगा that we are going to dilute or we are going to get the DNA in a concentration of 1 microgram in 1 microliter. क्योंकि जब हम स्टार्ट के ऊपर डीएनए एक्सट्रैक्ट करते हैं तो हमारे पास सैंपल कंसंट्रेशन जो है डिपेंडिंग अपॉन द स्टार्टिंग मटेरियल वेरी कर सकती है. सो बिफोर प्रोसीडिंग फर्दर टुवर्ड्स द सीक्वेंसिंग मेथड वी हैव टू गेट standard concentration of the DNA so that we can proceed with the subsequent steps easily with the defined volume which we may need for the sequencing step. So for this purpose, the first step is to take about 5 grams of the plasmid DNA in a 1.5 milliliter microfuge tube, which is Eppendorf tube, and the volume is made up to 50 माइक्रोलीटर. अब यहाँ पे अगर आप देखें हमारे पास जो 5 माइक्रोग्राम डीएनए है दैट इज इन 50 माइक्रोलीटर ऑफ द वॉल्यूम. जब इस प्रोसीजर के एंड पे हम पहुँचेंगे हमारे पास 5 माइक्रोग्राम जो डीएनए है दैट विल बी इन 5 माइक्रोलीटर वॉल्यूम. सो वी आर ड्यूरिंग दिस प्रोसीजर वी आर ऑल्सो गोइंग टू repurify the DNA so as to get rid of any kind of possible contamination which may have come during the extraction or the isolation of DNA. So the next step is going to be addition of 10 microliter of sodium hydroxide and EDTA which was freshly prepared, and then incubation at room temperature for 5 minutes. Once this 5-minute time period is over, we are going to add 30 microliter of ammonium acetate and the contents of the tubes are mixed by vertexing. Then 45 microliter of equilibrated phenol and is added into the tube. Again, vertex mixing is done so that all the contents are mixed thoroughly. The next step would be use of chloroform and isomyl alcohol mixture. 135 microliter is used, is added in the mixture, and vertex mixing is done. That vertex mixing is going to mix every content, and we will see that there are 2 phases. One is organic and the second one is a liquid phase. Aqueous phase is visible. So we have to separate these 2 phases by centrifugation. जैसा कि हमने ऑर्गेनिक एक्सट्रैक्शन ऑफ डीएनए मेथड में पढ़ा था कि कैसे हम ऑर्गेनिक फेज को अलग करते हैं और एक्वस फेज को अलग करते हैं और उसके बाद फिर हम अपने रिक्वायर्ड फेज को आइसोलेट सेपरेट करके डीएनए एक्सट्रैक्शन की तरफ जाते हैं. सो यहाँ पे हमें दोबारा से ऑर्गेनिक फेज को सेपरेट करना है फ्रॉम द एक्वस फेज बाय सेंट्रीफ्यूगेशन एट मैक्सिमम स्पीड फॉर 15 मिनट्स एट room temperature. Once the 2 phases are separate, we are going to carefully remove the upper aqueous phase to a fresh microfuge tube, and we will add 330 microliter of ice cold ethanol. This ice cold ethanol is responsible for precipitation of DNA. This precipitated DNA is going to come up or is going to be visible after 15 minutes incubation in ice ethanol bath. Mixing के बाद हम इस ट्यूब को ice ethanol bath में 15 मिनट के लिए रख देंगे. Ice cold ethanol is going to let the DNA precipitate, and that DNA pellet may be visible or invisible. So we have to recover this denatured DNA pellet by centrifugation for 15 minutes maximum speed at 4 degrees centigrade. अब इस centrifugation step के बाद a white spot may or may not be visible at the bottom of the Eppendorf tube in the form of a pellet. Even if it is not visible, we have to carefully remove the supernatant without disturbing the DNA pellet, which may be visible or which may not be visible. Then All the supernatant is gone. We have to recentrifuge it for a couple of seconds and remove any traces of ethanol. Once all the ethanol is removed, we will let this DNA pellet to dry in air, and later on we will add water in it to a concentration of 1 microgram per microliter. Once we get this purified DNA, then we are going to anneal the primers to this DNA, which is denatured, using 5 microgram of alkaline denatured plasmid DNA for each set of 4 DNA. That means 5 microgram of DNA is going to be split into 4 parts, 1.25 microgram, 1.25 microgram, and so on. And each 1.25 microgram DNA is going to be mixed up in the separate tube with the dideoxynucleotide triphosphate, one type, that is DDATP, the second tube DDGTP, the third tube DDTTP, and DDCTP, and so on. Then the annealing mixture is going to be added, which is containing alkaline denatured DNA, optional DMSO, or the enzyme reaction buffer. If the enzyme is sequinase, then sequinase reaction buffer. If this is the Taq polymerase, then the polymerase reaction buffer, then the sequencing primer, and water as per required volume. The volume of the water can be variable depending upon the total volume of the reaction mixture.